When we think about infectious diseases around the globe, we usually think of examples like tuberculosis, malaria, and AIDS. But what if I told you that over 250 million people in the world are living with a chronic form of this condition, and 600,000 die each year from it? You probably want to know what it is, right? Well, this condition is the most common liver disease in the world, and it's called hepatitis B. Hepatitis B is a potentially life-threatening liver infection caused by the hepatitis B virus, or HBV. It's a major global health problem which can cause chronic infection and puts people at high risk of death from cirrhosis and liver cancer. In terms of transmission, hepatitis B is primarily spread from mother to child at birth, sexual transmission, or misuse of contaminated needles. Most people do not experience any symptoms when newly infected. However, some patients experience acute illness with symptoms that last several weeks, including yellowing of both the skin and eyes, dark urine, extreme fatigue, nausea, vomiting, and abdominal pain. A small subset of people with acute hepatitis can develop acute liver failure, which can lead to death. In some others, hepatitis B virus can also cause chronic liver infection that can later develop into cirrhosis or liver cancer. In terms of prevention, a vaccine against hepatitis B is available. The World Health Organization recommends to all infants receive the hepatitis B vaccine within 24 hours after birth, followed by 2 or 3 more doses of the vaccine at least 4 weeks apart to complete the series. HBV vaccination is widely available. However, science does not always succeed, since nearly 10% of the general population fail to produce an immunologic response and develop immunity once vaccinated. This percentage is even greater among people with type 1 diabetes, reaching numbers as high as 50% of non-responders to the vaccine. Let's talk about type 1 diabetes, an autoimmune condition which represents 10% of diabetic patients. When you have type 1 diabetes, your body attacks the insulin-producing cells in the pancreas through the production of autoantibodies, so you can't produce insulin at all. The fundamental issue is that we all require insulin to live, as its function is critical to our survival. It allows glucose in your blood to enter all cells and follow our bodies. In fact, insulin could be analogous to the pipe that allows petrol to enter a car. When insulin isn't produced, glucose builds up in your bloodstream, leading to many body alterations and complications if not treated. So, what happens to a patient with type 1 diabetes and who responds to the hepatitis B vaccine? Why nearly half of these patients don't have antibodies against hepatitis B? Is there any characteristic in type 1 diabetic patients which could be associated with unresponsiveness to the HBV vaccine? These questions are intended to get answered with our project proposal, clinical and biochemical parameters in type 1 diabetes, predictors for HBV vaccine unresponsiveness. Let me explain a little bit what it's all about. In order to accomplish our objectives, it is of fundamental importance the patient enrollment. 230 patients are needed, so that will be gathered from 3 different hospitals in Barcelona. But how will this be done? Type 1 diabetic patients have many medical follow-ups, and in one of those appointments, the endocrinologist will hand them this leaflet with information about HBV, responsiveness to the vaccine, as well as the objectives, details about the methods and chronology, and future applications of the research. The physician will also shortly explain what the project consists of. In case of acceptance, a consent form will be handed to the participants to be signed, and a questionnaire will be filled by the physicians in order to collect some information for the clinical history. In the questionnaire, we will just ask for basic information about the participants, including age, sex, body mass index, smoking habits, and both abdominal and hip diameter, as well as parameters related with diabetes, such as onset of the disease, time of diagnosis, and autoantibodies levels at the moment of diagnosis. And finally, as obvious, the presence of vaccination against HBV must also be noted, as well both the time and the country of vaccination, as it wasn't until 1992 that vaccination was recommended for teenagers, and until 2002 the vaccination was started in Spain for the newborn. So far, it doesn't appear to be difficult, does it? Because what continues is along the same line. 36 months after the first visit, on the next analytical control, patients will only have to give a bit of saliva and an extra blood tube, in addition to the usual blood and urine sample, as to analyze the presence of HBV antibodies and diabetic mellitus HLA-raised alleles. Other conventional values will also be included: glycosylated hemoglobin levels, lipid profile, and microalbumin creatinine ratio. The HBV antibodies will be tested with an ELISA protocol, and people with levels under 10 milli-international units per millimeter will be considered as non-responders. Following the collection analysis of the samples, which will take about a year to be completed, staff will conduct a multivariate statistical analysis of the obtained data to see if there is any link between altered parameters in type 1 diabetes and non-response to hepatitis B vaccine. To do so, participants will be separated into two groups based on their levels of anti-hepatitis B antibodies. Responders type 1 diabetic patients when antibodies are 10 or higher, non-responders type 1 diabetic patients when antibodies are under 10. The two groups will be compared, and what do we expect to find? Well, we expect non-responders to have older age, higher body mass index, higher smoking rate, worse diabetic control, higher hemoglobin levels and more diabetic complications, including nephropathy, shown as higher microalbumin creatinine ratio, and retinopathy. We also believe that higher autoantibodies level diagnostic will be strongly correlated with unresponsiveness, as they are what make type 1 diabetes an autoimmune disease, as this group of diseases has been seen in some studies to be related to unresponsiveness, and of course, find the opposite in responders. So, what do we pretend with everything planned? If actually there is an association between certain levels and a vaccine non-response, the susceptible individuals to unresponsiveness will regularly be checked for hepatitis B antibodies and given a booster vaccine in case of levels falling below 10. Basically, the protocol will be as follows. Once someone is diagnosed with diabetes mellitus, parameters associated with unresponsiveness to HBV vaccine will be assessed. If they are present, a serology test for antibodies anti-hepatitis B will be carried out, and in case of those being under 10, a booster dose of the vaccine will be injected. In case of the answer of either of these steps being negative, then a regular endocrine control will be done. But this doesn't end here. It is obvious that many further studies could follow the proposal exposed by studying the effectiveness of the booster dose, comparing HLA risk levels not only in diabetic patients but also in the general population to check for the implications that having the disease has, studying the possible etiology of the unresponsiveness, etc. So here is a summary of the steps to follow: patient enrollment, sample and data collection, statistical analysis, obtention of the results, and protocol development. All in all, what we aim to achieve is a reduction of the number of people exposed to hepatitis B virus, in particular, people with type 1 diabetes, as they are a group exposed to a greater extent. Hepatitis B is a life-threatening infection which can cause thousands of deaths, and it's exactly for this reason why it is essential not to underestimate it, and to put a great effort into reducing its spread, making the world a little safer for all. Thank you for your attention. We honestly hope this was a useful and interesting video. We'd like to thank all the professional help we have received from Judy Garcia, Manuel Pastor, Nomi Robles, Juan Jose Chiaron, and especially your tutor, Andres Ozaita, for the support given at all times.